All right, I'll start by introducing myself. My name is Stephen MacKinnon. I'm the VP of Research and Development at Cyclica. I guess a little bit about my background. I started off with a bachelor's degree in computational biochemistry. I did a PhD in the same subject. And in late 2013, early 2014, I joined very early stage Cyclica, where I'm pretty much doing the same thing still today. So very linear, very direct trajectory on that one. Yeah, so my role's kind of evolved. The company that I work for, Cyclica, has grown over the last 6 years. I started off very much being a developer, doing a lot of code work, and now my role is more in terms of coordinating between multiple different teams and establishing a plan for development and executing on that plan. So, and I'll speak a little bit more about what Cyclica does kind of over the next little few slides. I'm also taking a look, I'm seeing these polls in here right now, and I'm seeing the most popular, what types of talks are you interested in? Technology. I'm kind of feeling bad now that I wasn't more technical with the slides. I don't think that there's any rock curves or anything in there. So we'll see. All right. A lot of the talks today are going to be in the space of computational drug discovery. It's a space that I find particularly interesting. It has a very long background. People have been using computers for the sake of doing simulations of chemistry and trying to design drugs for quite some time now. And again, it's not at all a new thing. I just got to reduce this so that all right. So it's not at all a new thing. People have been looking at the 3D structures of proteins and the 3D structures of drugs and trying to fit things in all the way back into the 1970s and 1980s, where there were big promises of computational drug discovery, really changing the universe of how drugs are discovered. And even the idea of using machine learning in chemistry. So this is a publication. I like to point this one out because it's as old as I am, April 1984. And it in itself is a review of people using machine learning in the field of drug design and what kind of models people were developing in the 1960s and 1970s, what were the shape of those models. And you see a lot of the same keywords in there: structure-activity relationships, additivity models, multiple parameter analysis, pattern recognition. A lot of the topics that are really trendy today, kind of retro there, seeing something from 36 years ago, I guess. And when people, at least in textbooks, when people talk about computer-aided drug design, the paradigm is often breaking things down into two different groups. Are things 3D simulation oriented, meaning, are you trying to replicate physics and trying to look at molecules and simulate time and how molecules interact with one another, or using machine learning to find these relationships or map these functions that are a little bit more obscure than those that can be explained by straight up physics in a way to predict and come up with new things. So this is really kind of a long-standing paradigm of the way in which drug design is talked about, at least in the early stages of identifying new drugs. And well, there's a lot of attention in this space, particularly recently, and particularly with smaller companies. So, back in the 90s, 2000s, the pharma discovery system was very much dominated by large entities, whereas now with the AI, a lot of it is coming through smaller groups. So groups, startups like us, startups like the rest of the groups here presenting tonight. And there's a lot of effort being put into this. An ongoing blog that gets updated every few months, written by Simon Smith, who works out at BenchSci, which I believe the next presentation, somebody will be presenting from BenchSci, kind of lists those out, the 230 startups using artificial intelligence for drug discovery. And other people kind of tracking that growth, 2017 to 2018, investment rising fourfold year over year over a period of time. So there is a lot of interest generated in the space. And, hopefully over the next few slides, I can kind of help and explain why that is. This is a figure I think that a lot of people in this space may be familiar with. It's how many drug candidates people look through when they're going through the early stages of drug discovery, and what is the attrition as it goes through different phases. As it goes into preclinical research, as it goes into phase 1, phase 2, phase 3 clinical trials. And that is both attrition in terms of programs being lost, molecules being lost, and money to some sense. So this is good in terms of just getting an idea at a very high level. But it's not as simple as just saying that computers are going to come and help jump things from one phase in the drug discovery all the way into approved medicines, right? So the reason why that is this in itself is a very narrow image of what the drug discovery pipeline actually looks like. So here's another graph that kind of describes, instead of just saying preclinical phase 1, phase 2, phase 3 for drug design, this is a map of all the different steps that go into a conventional drug design product, right? So what is that product roadmap, if you will, of a drug? And it's too big to even fit on the screen. I'm sure a lot of you are probably zooming in right now and trying to look. I actually had to split this slide up into two so that the first half of the slide was one part and the second half of the slide was the other part. And again, it's not really, this isn't really a matter of so one thing that I wanted to kind of convey here is that this is a very complex system, that there are a lot of, the reality is a lot more complicated than just looking at a funnel and saying that AI can help move things from one side to the other. There's a lot of different steps, right? This pharma product roadmap is very complicated. There's a lot of these interconnected processes and many bottlenecks, right? And these bottlenecks might actually change from research program to research program. In some cases, it might be a step right here that's blocking things up, and another one, it might be a step right there. So the reality of where somebody can come in and introduce efficiencies to make things work better, right? So when we go back to that slide earlier on, 230 companies in this space, well, there's a lot to do. There's a lot of steps that could be improved. Data is very important at every single one of these stages. And you could pick a handful of 10 companies on that list, and chances are none of them are really doing the same things and offering the same value at the same point in the process. So one of the frameworks that I like to think about when kind of evaluating the potential of AI or a new ML solution in a particular space actually comes from the education sector. This is a framework that people use to describe the impact of a technology on education, but specifically on education. But I think that it's a really interesting thing to think about outside of education, just everywhere, really. Like a new technology coming in, what is the impact of that particular technology? What is the hierarchy of different impacts of a technology, right? And so these are 4 different levels that you can conceptually break down the impact of a new technology on a problem as a whole. Is that new technology just acting as a direct substitution of something that already exists? Is it somehow augmenting that process, right? Is it a direct substitute, or does it have a functional improvement as well? So those would be under the bit of enhancement. Maybe you're doing in an existing process, you're just doing it faster. Or maybe you're adding new insights that didn't exist in the previous process before. But then the higher order impacts would lend themselves to modification and redefinition, right? So are you actually redefining, are you actually modifying the problem or redefining what the problem is altogether, right? So if we think about that network that we were just showing before, these enhancements would be working within that existing framework, that existing network below, and addressing the same inputs and outputs, right? So you find one of the nodes and one of the edges in that graph, and you do something to improve that. When we're thinking about more transformative technologies, we're thinking potentially about rewiring that network, not working within the existing, framework or the existing pipeline, but trying to reconsider it altogether. And at least personally, those are the tasks that interest me more, that grab my attention. And if we think about drug design process, going into example opportunities for improvement, well, why are there so many companies out there? Why are there so many opportunities? If we think even before we go, or if we think about the high-level questions that people might be asking is, what kind of proteins can I target to get a particular therapeutic effect? You start with the disease out there. There's a lot of diseases. I looked up a database recently. There's over 11,000 named diseases. So there's never a lack of work in this particular space. There's always people that we can help. There's always therapies that can be improved. So within that target identifications, some other questions that people might be asking is, well, what kind of models can I use to represent the disease? Because we're not testing directly on human here. We normally have other types of systems like animals or cell models in order to test if a particular change is giving us a desirable therapeutic effect. And then there's the act of actually finding new molecules that could impart that therapeutic effect. So which molecules can alter the target protein or the target biological activity? Can we fine-tune those to be more active at a lower dosage? Can we know in advance which ones will be safe or non-toxic at the next stage? Within clinical trials, how can we recruit the right people? Can we find new markers that will track whether the drug is working or not? Can we segregate populations to see in advance maybe there's a subpopulation in which the drug would be more successful? And then, post-market, can we find new uses for existing drugs? And could we decide amongst a class of drugs which drug is the best for a given individual? So there's a lot of company in this space. There's a lot of attention in this space, mainly because there's a lot of problems and there's a lot of innovation opportunities. And it's an area that I'm quite passionate about and that I really kind of enjoy working in. So I hope that was a decent primer for people out there who are not necessarily in this space. We just kind of jump into what we do specifically at Cyclica. So we look at small molecules, and what kind of got our attention at the beginning of this is that these small molecules will interact with anywhere from 30 to 300 separate off targets. So even, like, as much as you might be designing a molecule to have an effect on one given protein, chances are it's physically interacting with tens, if not hundreds of other things in a cell. And well, what are those effects, right? Clinical trials and testing, preclinical and clinical trials, is all about crossing your fingers and hoping that those drugs don't do undesirable things. And a lot of attention has, gone into this field of off-target effects more and more in recent times, to the point where they're even discovering, within clinical trials, that the reason why drugs are effective, drugs are effective for the wrong reasons sometimes, and people aren't even realizing that until the drugs are kind of out there in the clinic. So the technology that we built, at least one of the primary technologies that we do to address this, we call MatchMaker. That's to predict interactions between proteins and ligand binding sites. And this is to kind of augment or even replace the docking simulations that people were using in the past with target-based approaches. Deep learning models trained on millions of drug-target interactions, combining both structural and experimental data. We find that it's very accurate, very generalizable, and definitely superior to the simulations, which were more time-consuming that we had done in the past. And we have our kind of proprietary data engineering and featurization engines related to protein structures in order to augment these databases with the right type of information in order to make better predictions. So validation, we do pretty well at being able to identify whether a given drug and a given protein would interact. But we set that to a more challenging task of saying, you know, for this particular drug, what are all the proteins it would interact with? And, you know, that's kind of analogous to, you know, at first you might be doing, trying to identify numbers, handwriting numbers, right? You know, that'd be one part of it. Can you do that reliably enough to do math with that is kind of the next analogy, right? Can you do something higher order with those types of predictions? The problems that we tend to focus on are very pragmatic. They're very applied in that bit of saying, these are real world problems. This one in particular, we're using it's a new approach that we call filter transfer learning. It's a way that we use transfer learning to basically train at different tiers of data confidence in order to boost our model performance. And we got some data to show that that strategy works pretty well. So improved neural net-based training with noisy data without compromising contributions from high-quality data points. Instead of just setting your threshold of saying, you know, this data is good, this data is bad, we're saying, let's use the positive data and let's use it more effectively than we would use noisier data, or data in which we can quantify and we know is noisier. Another similar ML, these are all publications, so we've got little snapshots. These are the types of problems that we work on, and very open about publishing these as we go. Another one that we're working on is POEM. It's another type of machine learning classifier, not deep learning based, in which if you have multiple ways of describing similarity between two different items, you could take that to an advantage. You could take that into account and create classifiers that will use multiple different definitions of similarity between objects to make their predictions without subjecting yourself to biases that you would normally do by blowing out features, your feature space. So it's a fairly accurate way of doing these types of predictions with a low risk of overfitting. And it's very ideal for automated model building. So if you want to use basically machine learning as a data mining technique and basically to see, you know, hey, is there enough information contained within these data sets to produce a model? And if you want to do that systematically across a large number of things, it's an ideal type of classifier to do tasks like that. Ligand design. So we developed at least our core engine around being able to say, can we list all of the proteins that a given drug is binding? Ligand design is about using that to create or design better drugs. We'll screen either fixed libraries of compounds, so like a library of 1 million molecules. We'll screen it all by all against the proteome and say, you know, these are the ones that have ideal profiles that would be good for a candidate drug. Semi-generative, in which we would take pieces of one molecule, recombine them with pieces of another, so that we're expanding the amount of chemical space that we can cover, but in a way that, you know, chemists can still go and synthesize them, or fully generative, in which we would just imagine new molecules and hope that chemists can build them afterwards. Another thing that we've kind of contributed recently for people that might be interested in is Deriver. It's an pip installable open source tool for people to make changes to molecules in a very guided way. So if you have any given model for drug activity for a given drug, you can apply this to sample chemical space or to navigate it and try to find molecules that satisfy the model that you've built and design new drugs yourself. At the very end of it, when we design molecules, we provide a platform for people to kind of interrogate that, because with prediction comes a bit of, it's very important that scientists integrate their own knowledge into systems, and by providing an interactive web platform is the way in which we help bring that information, that knowledge and that experience of the end user scientist, and allow them to interface directly with data. That's really important for us. The visualization, interpretation, and that ability to for scientists to use what they've learned in refining their predictions. I think the last effort that I want to talk about is PolypharmDB. This is, as I mentioned, one of the things that we do is we cross-reference, so we screen all human proteins against drug libraries. We screen libraries of known molecules. So these would be like libraries of FDA-approved molecules or molecules that have gone through clinical trial. And then we could just cross-reference those for targets in order to have some very quick hypotheses to test for new medicines and repurposing.
Yeah, thanks so much for that, Stephen. And a physical applause from me, and you also get a cheeky little hand clap emoji as well. So yeah, thanks so much for taking the time. I have a question just off the top of my head. When it comes to kind of drug discovery, and I mean in the time that we're in at the moment, when we have literally a deadly virus going around like COVID, what's the process that Cyclica will take? I mean, are you guys and girls, I should say, is your team actively involved in any of the current antiviruses or vaccines for COVID or any other types of disease? And if you can kind of tell us a little bit about the process of that.
Right. So the computational groups were amongst the first to start, putting out predictions and so forth. That last article that I kind of linked, that was PolypharmDB. We were looking at things back in January and put this resource together to specifically for COVID-19 to identify some repurposing opportunities. We've partnered with a number of different Canadian and international groups. So without being the deep experts in the science on, I'm not a virologist, we know how to make these types of predictions. There's a lot of science that goes into saying what is the right thing that we should be targeting. What is the right thing we should be targeting on the human side, on the virus side? So a number of different groups internationally had different ideas as to what the right course of action would be in multiple different lines of defense, right? So when it comes to the coronavirus, there's people who are coming up with vaccines, people who are coming up with antibody drugs. And when it comes to small molecule drugs specifically, there's those that act directly on the virus, and then there's those that act on humans to try to alleviate some of the effects of the virus, right? So we're working with groups on a number of different fronts with that. We've provided them with data generated from our predictions, and it's a combination of, whether that biological hypothesis was correct as to, is this something that will have an effect in reducing the severity of cases, for instance, and on that predictive bit of saying, will this drug actually interact? On top of that, we're looking at the first line of compounds that went through are the ones that were already through the FDA clinical trials. That's kind of one arm. A second arm there would be new molecules that might not have been approved for anything, but might have been preclinical molecules that companies have already built that they basically have a fast track to, or completely new molecules altogether, right? And we can see that there's actually efforts. This pandemic is very different from anything we've seen in the past, just because all of these things are happening simultaneously and in ways that are way faster than people have seen before. Even the de novo drug design, there's some progress that we're seeing internationally of people who are coming up with new molecules right away that are effective versus timelines that would have been years before. So there's a lot of hope. The second and third lines of defense outside of the things that we've already seen be approved or going through clinical trial now be very useful if new strains emerge in the future, because as grim as that might be, that's definitely a possibility of some of these therapies becoming ineffective because the virus changes over time.
Sure.
And it's like the influenza changes during different flu seasons. We might be facing different waves in the future.
Yeah, sure. We'll jump onto a couple of other questions there before we wrap up your talk there, Stephen. So can you elaborate on the deep learning aspects of ligand design and ligand express and MatchMaker?
Yeah, so the deep learning component is MatchMaker specifically, and that's where we take a representation, a feature-based representation of ligands, and a feature-based representation of proteins, specifically the binding site of a protein, rather than the whole of a protein. Proteins can be hundreds of, if not thousands of times larger than the small molecule drug. So, when we come to finding the right way to describe what a protein is for the sake of making this prediction, we want to focus on the elements that we know are the most important. So we have our kind of internal way of representing what a drug is and what a protein is. And we use combinations of different data sources in order to start training the models. So we'll find known interactions from a combination of different databases. We have an internal pipeline to take, 20 million or so of these known interactions from public databases and map them onto protein structures so that we can get a better representation of, the actual chemistry that's happening around and ways to describe the pockets. And then, we'll kind of train on all the positives. We'll randomize and basically shuffle in order to get negatives. And that's where the filter transfer learning comes into play. We can kind of quantify how well or how much we trust a given data point that we have, how well we trust that that data point is accurate based off of the source that we found that piece of information, how much we trust that that information is accurate based off how well we think we're mapping it onto the protein structure. And we use that to our advantage in training the model, right? And that filter transfer learning is a good article there that I think it demonstrates it doesn't fully describe MatchMaker in the way that we use it commercially, but some of the premise there as to how we think about data quality and how we integrate that into our training processes.
Awesome. Thanks so much for that, your question, Salim, and the answer there, Stephen. So we've got two more questions, but if we can kind of wrap these up pretty quickly because we are slightly just a smidgen behind time, but I do want to get to all the questions, certainly. So question from Reza Jafar. Thanks for the presentation. How do you get access to the training data for the drug proteins? There's
a lot of public sources. In our commercial engine, we have a combination of public and private sources feeding into it. But in terms of public sources, just as a resource to get started, I think there's Stitch and Kemble. There's a lot of open databases that are just for that. Whenever somebody releases a publication, the journals require people to take the source data and deposit it into a database. And that's where those databases are and kind of why they exist. So if we go back to that filter transfer learning paper a few slides back, this one actually comes with, we reference a a GitHub account here with some code snippets. And within that code snippets is a very good, simple way to kind of get started if you're interested in drug target interaction predictions. You know, a source database and how you basically take information from that source database to come up with some basic features. So it's there's a nice little code base there that provides a toy model for people to play around with.
Excellent. And the last question we have for you is from Shilpi. Hi, Stephen. There's a few questions in here. So if the drug discovery process takes so long and it's really difficult to zero down on a few molecules, how come COVID vaccine is so fast-paced?
Okay, so the vaccine is a little bit different. And there's also this effect that everybody is working on this right now, right? So if you're running an internal program, you go through maybe 20 iterations of trial and error, and you know, consecutively, and that could be a 2 or 3 year process for any given step. But when everybody is working on this globally, there's some trial and error happening just by virtue of different groups doing different things, right? So even if the success rate for something like a vaccine might be 1 in 50, if you've got 300 groups globally working on it, there's a few that are going to pop up as being successful right away. So there's definitely a number games going into it. This really is one of the things that turned everything on its head and got going really quick for both the vaccine and compounds. I think the more successful compounds that we saw the first kind of wave of so the very first wave of small molecule drugs that people were associating with were chosen because there was already some evidence that they worked on viruses somehow. Even the anti-malaria drugs that people were talking about, there's some papers that were linking those to the original coronavirus SARS outbreak back in 2003. So people had a reason to specifically look at those as having been related to viruses in general. And almost anything that's ever been linked to a virus was tested within the first 2 months, given the number of groups working on this globally. The next wave are the things that don't have any immediate connection to viruses directly. And then the wave after that would be things that like non-obvious connections. And then the wave after that would be completely new medicines from the ground up. And all of that is happening pretty quick.